Innate immunity is the immunity you are born with, and adaptive immunity, also called specific immunity, is acquired through exposure to pathogens throughout your lifetime. Pathogens can be bacteria, viruses, or parasites. Basically, anything that disrupts homeostasis. The immune system is composed of millions of molecules and cells working to destroy anything identified as not being part of you. The foot soldiers in this war on microbes are the white blood cells. White blood cells are large, translucent blood cells with an arsenal of organelles. All white blood cells are leukocytes. Leukocytes form in the bone marrow. After their formation, they follow different pre-programmed destinies to dispose of microbes by engulfing them, lysing them, or synthesizing products to help destroy them. White blood cells can be divided into different subsets based on the type of immunity that is triggered for defense. Monocytes, which become macrophages, neutrophils, and eosinophils, are all considered phagocytes. Phagocytes destroy pathogens by engulfing them in vesicles, internalizing them, breaking them down, and spitting out the remains. Another important subset of white blood cells is the lymphocytes, which include B cells, T cells, and natural killer cells. Lymphocytes are more subtle assassins. They poke holes in cells, injecting enzymes that kill the microbe, or they produce antibodies that smother the intruder, targeting it for destruction. Phagocytes and natural killer cells are important members of innate immunity, while B cells and T cells are active members of specific immunity. Innate immunity is your first line of defense. This defense begins with your skin and mucous membranes. Innate immunity relies on two broad categories of cells for defense: phagocytes and natural killer cells. As we discussed, phagocytes destroy pathogens by engulfing them in vesicles, after which they break them down and spit out the remains. Natural killer, or NK, cells are derived from lymphocytes. They migrate over all the tissues of the body looking for and destroying abnormal cells like cancer cells and virally infected cells. The NK cells can recognize these cells as different because they lack the appropriate signals identifying the cell as being you. This lack of you traits is usually in the form of a tag, such as a specific cell membrane receptor or the presence of unidentified sugars that belong to the pathogen. Unlike phagocytes that engulf their prey, NK cells come in direct contact with the target cell, poke holes in the membrane, and inject it with toxic chemicals. This type of attack is very similar to another lymphocyte we will talk about later called the T cell. NK cells also secrete potent chemicals that act like flare guns to augment the inflammatory response. This flare gun signals macrophages, other lymphocytes, and some of the chemical defenses to help combat the infection. The arsenal of chemical weapons includes the inflammatory response, the secretion of antimicrobial proteins, and fever. The inflammatory response kicks into gear when you suffer from some physical injury, such as getting kicked. It also becomes activated by injury from intense heat, irritating chemicals, or infection from viruses, bacteria, or fungi. Inflammation begins with a warning siren made up of chemicals secreted by the injured tissues. These chemicals act as a homing beacon for macrophages and mast cells to migrate to the injury site. Macrophages start engulfing bacteria and debris, while mast cells secrete histamine. Histamine is a very potent inflammatory chemical that causes the symptoms we associate with allergies. Histamine causes the surrounding arterioles to dilate. This brings warm blood to the area and increases the local temperature. In addition to this, the histamine increases permeability of the local capillaries to promote exudation, the leaking of capillary fluid into the tissues, carrying antibodies and clotting factors with it. This is what causes the swelling. And the swelling, in turn, presses on nerves, causing the pain. If the injury is badly infected, pus can develop. This creamy yellow substance is a mixture of dead white blood cells, ruined tissue, and living and dead pathogens. If the area is very badly infected, it can be walled off from the rest of the body by scar tissue. The scar tissue is made from collagen fibers and can form an abscess. At this point, the pus and fluid buildup within the abscess will have to be drained before the injured area will heal properly. Besides the inflammatory response, innate immunity also includes antimicrobial proteins. These antimicrobial proteins include the interferons and complement proteins. Interferons are proteins secreted by infected cells in a last-ditch effort to save adjacent uninfected cells from viral attack. Interferons diffuse into adjacent cells and cause the still healthy cell to synthesize proteins that stop viral protein synthesis and degrade the viral RNA. The complement system, usually just called complement, includes a group of more than 20 plasma proteins that circulate in an inactive state. Their function is to jack up the response by the immune system to its highest active status. It does this through chemical mediators that enhance every aspect of immunity, hence the name complement. Complement can be activated either by antibodies or when certain complement factors interact with microbial molecules and target them for destruction. The final component of innate immunity is fever. While inflammation is a localized response, fever is a systemic response. That means it affects your whole body. Fever usually occurs if the infection is widespread and internal. Fever is an increase in the body temperature that occurs when your internal thermostat, regulated in your hypothalamus, is reset to a higher point. The reason your thermostat gets reset is because leukocytes and macrophages secrete chemicals called pyrogens. Low and moderate fever is an effective immune response. It speeds up the body's metabolic rate, which in turn accelerates the repair process. Immunity is your body's defense against pathogens. There are two types of immunity, innate and adaptive. Innate immunity is the immunity you are born with and includes both physical barriers, such as the skin, and internal defenses. Internal defenses include cellular defenses performed by phagocytes and natural killer cells, as well as inflammation, antimicrobial proteins, and fever. Adaptive immunity is the body's specific immunity. Unlike your innate immunity, which is nonspecific and attacks anything it recognizes as foreign, damaged, or unknown, adaptive immunity is very choosy. Adaptive immunity will only attack specific threats. Adaptive immunity is carried out by two types of lymphocytes, the B cells and the T cells. Like all blood cells, the lymphocytes first develop in the bone marrow. B cells stay and mature there. Immature T cells migrate to the thymus where they mature. These lymphocytes become immunocompetent and have to be self-tolerant. In other words, these cells have to be able to evoke an immune response and make sure that response isn't against your own normal cells. Adaptive immunity has three underlying traits. First, it is specific. It recognizes and attacks only specific pathogens. Secondly, it is systemic. This is a widespread reaction not limited to a localized region of the body. Lastly, adaptive immunity has memory. In many cases, once you are exposed to a virus, your body remembers and you become resistant to reinfection. Adaptive immunity has two components, humoral immunity and cellular or cell-mediated immunity. Humoral immunity produces antibodies. Cell-mediated immunity relies on a particular lymphocyte called a T cell rather than antibodies to defend the body. Both of these components work by recognizing antigens. Antigens are cell surface proteins. You as an individual have a unique set of self-antigens called your major histocompatibility complex, or MHC. However, when cells are infected by a pathogen or are abnormal, like a cancer cell, non-self antigens are presented. The change can be initiated by the infectious agent or because the cell no longer responds to normal control signals. It is these changed cells that the humoral and cell-mediated immune processes recognize and destroy. There are two classes of MHC proteins. Class 1 MHC proteins are on all the cells of your body. Class II MHC proteins are only found on specific cells of the adaptive immune response. The B cells are formed in the red bone marrow, where they mature and become immunocompetent. These are the cells responsible for producing antibodies against foreign antigens. Once a B cell is exposed to a foreign antigen, it becomes activated and undergoes cell division. One of the daughter cells becomes a memory cell. That is why you can be resistant to reinfection of, say, chickenpox once you have been exposed. These memory cells may live up to 20 years or longer. The other daughter cell has a different fate. It will continue to divide and produce an army of B cells. Each B cell will go on to produce antibodies that are secreted into general circulation to seek out and destroy the antigen they were exposed to. Antibodies mark the target cells for destruction by causing them to clump together, smothering their surfaces, acting like beacons for complement proteins and phagocytes, and causing toxic cell byproducts to precipitate out of solution. But there are limitations to their defenses. B cells can only detect an obvious threat, like bacteria floating around in your blood. They can't detect viruses and bacteria that infiltrate the cells. For this, another defense is mounted by the cell-mediated arm of immunity. Cell-mediated immunity has a more direct approach. T cells are formed in the red bone marrow and then migrate to the thymus where they become immunocompetent. T cells recognize infected or abnormal cells differently than B cells. They recognize antigens that have been processed into fragments and sent to the cell surface. Once it detects one of these fragments, the T cell binds to it and becomes active. The T cell then divides like the B cell, with one daughter cell becoming a memory T cell and the other the cytotoxic T cell that targets cells with that antigen fragment for destruction, up close and personal. The cytotoxic T cell binds to the foreign cell, pokes holes in it, injects enzymes and toxins into it, and breaks up the cell membrane. The B cells are responsible for the humoral response. This response involves identification of foreign antigens. This activates the B cells to produce antibodies specific for the targeted antigen. They attack and destroy these antigens or antigen-producing cells by clumping, lysing, and precipitating them out of the blood. So humoral immunity can be considered an indirect attack on pathogens by the B cells. In contrast, the T cells initiate a specific immunity response in which the T cell is presented with antigen fragments that cause the T cell to become immunocompetent. Once this occurs, the T cells destroy the unwanted cell by poking holes in the membrane and injecting enzymes to break the targeted cell into pieces. With adaptive immunity, the first time the B and T cells are exposed to a new antigen, the response is slow. So you get really sick. This is called the primary response. The second time you are exposed, the B and T memory cells have a record of this antigen or antigens like it. So they can respond and divide really quickly, acting like a first strike against the developing infection. This is called the secondary response. So when you are exposed to chickenpox the first time, you get an intense illness. But you don't get it a second time because your B and T cells can very quickly replicate and destroy the pathogen. That is how vaccines work. A vaccine initiates a primary response at low levels, deliberately introducing the B cells to the antigens. The B cells produce antibodies against the antigens and form a memory of these antigens. Then the next time you get infected, the B cells can respond quickly because they already know to attack the pathogen. It is specific and self-tolerant. B and T cells are the major players. Upon exposure to an unknown antigen, they produce memory cells. B cells produce antibodies, which mount an indirect attack on the infected cells and non-cellular pathogens. T cells attack directly to kill the cells by poking holes into them and injecting cytotoxic compounds.